Hi everyone, my name's Ray, and I've completed my project along with my mentor Anish on summarizing TREM2 signaling pathways in relation to Alzheimer's disease. So, for starters, let's see what Alzheimer's disease is. So, Alzheimer's disease is a progressive neurodegenerative condition that leads to cognitive decline, and this contributes to around 60 to 70% cases of dementia across the globe. Symptoms of Alzheimer's disease are memory loss, inability to learn, hallucinations, and paranoia. So what essentially happens in Alzheimer's disease is that the cerebral cortex that is responsible for language and information processing shrivels up, and this leads to impaired thinking and planning. And the ventricles of the brain are filled with CSF, that is cerebrospinal fluid, which leads to swelling. And hippocampus, which is responsible for forming new memories, shrinks severely due to the neuronal damage that's caused in Alzheimer's disease. So Alzheimer's disease is, the pathogenesis of Alzheimer's disease is complicated by an interplay of aging, environmental toxins, genetic factors, etc. So there are essentially two types of Alzheimer's disease, the first one being early onset Alzheimer's disease or familial Alzheimer's disease, which accounts for around 5 to 10% of all Alzheimer's cases and mainly affects people below the age of 65. The potential pathogenic causes for early onset Alzheimer's disease are mutations in amyloid beta precursor protein and presenil 1 and 2. The second one is late onset Alzheimer's disease, also known as sporadic Alzheimer's disease. This accounts for majority of the Alzheimer's cases, and it mainly affects people above the age of 60. While there are numerous genetic factors that are present that are involved in load pathogenesis, one of the strongest genetic risk factor that has been observed is apolipoprotein E, which consists of 3 different alleles, E2, E3, and E4, with E2 being least involved in Alzheimer's and E4 having the highest amount of involvement. So hallmarks of Alzheimer's disease, there are essentially 2 pathological hallmarks of Alzheimer's disease: the extracellular beta amyloid deposits and intracellular neurofibrillary tangles of hyperphosphorylated tau protein. These two combined lead to synaptic and neuronal loss that causes atrophy in affected areas of the brain. So while Alzheimer's was initially considered to be a cell-autonomous neurodegenerative condition, which essentially means that the hallmarks are not affected by the cell-to-cell interactions in the brain, neuroinflammation was observed in recent Alzheimer's studies, and this and recent accumulating genetic and functional evidence also indicates the relation of Alzheimer's pathogenesis with innate immunity. Role of microglia in Alzheimer's disease. So microglia is essentially macrophages that are present in the central nervous system that act as the first and main line of defense in the brain. So what essentially happens is its relation to Alzheimer's. In case of Alzheimer's, there is, due to an abnormal increase in A beta production, A beta accumulation in the brain, there is an abnormal amount of increase of NLRP3 inflammation, which leads to abnormal cytokine production. This, combined with aging, leads to impaired amyloid beta clearance, and this sort of perpetuates a cycle of neurotoxicity in the brain. Eventually leads to neuronal death. Relation to tau pathology. So endogenous tau is shown to have role in neuronal transportation due to its involvement in assemblage and stabilization of microtubules. So what essentially happens is when this endogenous tau detaches from the microtubules, their abnormal microglial activation and pro-inflammatory cytokine production is observed, which subsequently leads to hyperphosphorylation. This drives an overproduction of pathogenic tau and perpetuates the cycle of neurotoxicity. So what essentially microglial pattern recognition receptors are is the ones that sort of notice any abnormal activities that go on in the brain. And one of the microglial pattern recognition receptors that is involved in Alzheimer's is TREM2. So what is TREM2? TREM2 is triggering receptor expressed in myeloid cell 2, which is an innate immunity receptor observed on the surface of microglia in the brain. It is encoded by the TREM2 gene located in human chromosome 62.1.1. The TREM2 signals are associated with ThyroB or DAP12 complex through transmembrane domains. The interaction of TREM2 and DAP12 essentially mediates several microglial neuroinflammatory and phagocytotic responses, such as debris clearance and cytokine production and release. The TREM2 gene mutations have been shown to be involved in pathogenesis of neurodegenerative diseases such as Nasu-Hakola, Alzheimer's, Parkinson's, and ALS. TREM2 signaling pathway. So there are three kinds of TREM2 signaling pathway. The first one being extracellular sensor domain, which contains an immunoglobulin domain that binds to pathogen-associated molecular patterns, damage-associated molecular patterns, cell debris, lipids, and apolipoproteins. The transmembrane domain associates itself with DAP12 and forms the TREM2-DAP12 complex. And TREM2-ICD is a short cytoplasmic tail which is released via gamma secretase cleavage, and its function as of yet remains. So the combination of TREM2 and DAP12 sort of activates a cascade of intracellular signaling pathways events. So DAP12 consists of immunoreceptor tyrosine-based activation motif, also known as ITAM. This, along with tyrosine residues, become phosphorylated when TREM2 binds to a particular ligand. This then recruits spleen tyrosine kinase that activates downstream signaling, along with several molecules such as guanine, nucleotide exchange factors, etc. Depending on the context and the molecules that are then recruited, the TREM2 DAP12 complex affects several microglial functions, as we have seen before. So the TREM2 variants that are present in Alzheimer's disease. So mutations in TREM2 gene result in production of mutant TREM2 proteins that differ from normal proteins only by a single amino acid. It has been observed that the R478 mutation of TREM2 significantly increases load risk, and it is shown to sort of limit TREM2-mediated sensing of lipids in microglia. It has also been hypothesized that apart from R478 mutation, there might be other mutations that might be involved in the Alzheimer's pathogenesis. A study conducted observed 16 non-synonymous variants, 6 of which were not identified in previous Alzheimer's studies, and three of which were expressed in human brain, along with one which encoded for soluble form of TREM2, which is shown to have protective function in CSF and also is shown to be involved in mediating pro-inflammatory responses in the brain. So this is a link between TREM2 signaling and Alzheimer's disease. So for first one, its relation to amyloid beta, there's a reduced uptake of amyloid beta in the brain in case of TREM2 deficiency, and there is an observed multifaceted microglial dysfunction that has been observed. It is hypothesized that this might stem from impaired mTOR or mechanistic target of rapamycin signaling, which is essentially which faces increased activation in case of A-beta accumulation. In case of tau pathology, there is increased phosphorylation and aggregation of tau proteins. In case of TREM2 deficiency, TREM2 is shown to enhance the pathological effects of macroglial activated microglia on tau, accumulated tau in case of Alzheimer's by suppressing the inflammatory response produced. Its relation to ApoE is that it is shown to have increased phagocytosis of apoptic neurons via TREM2 signaling pathway. TREM2 deficient microglia is also shown to have reduced uptake of amyloid beta complex, amyloid beta and ApoE complex, and amyloid beta LDL complexes. For conclusion, so over the past few years, several genetic studies have confirmed the involvement of numerous genetic factors in pathogenesis of Alzheimer's disease. And amongst all of these genetic factors, TREM2 and its variants, their impact on Alzheimer's has been observed. In this project, my mentor and I have attempted to summarize the importance of TREM2 functions and signaling pathways in relation to Alzheimer's pathogenesis. We also discussed the relation of TREM2 signaling pathway and AD hallmarks, and explore the relation between ApoE and TREM2 and the joint impact on Alzheimer's pathogenesis. So despite the countless studies that have explored the relation between TREM2 and Alzheimer's and the role of TREM2 signaling pathway in Alzheimer's therapeutics, there still remains a sort of knowledge gap with respect to how this dual role of TREM2 and its impact on TREM2 microglial functions can affect Alzheimer's and can also aid in therapeutics of Alzheimer's. Despite all of the research that has been going on and the interest that's shown towards the involvement of TREM2 signaling pathway in Alzheimer's therapeutics, there still remains a lingering doubt as to how and when should the TREM2 signaling pathway should be stimulated in Alzheimer's with respect to therapeutics, and whether this will help in sort of help the non-carrier Alzheimer's patient who account for majority of the Alzheimer's cases. So one of the ways that this knowledge gap can be filled is by using techniques such as CRISPR, for example, CRISPR and mathematical modeling. For example, McCade et al. Has shown via CRISPR system that knockout TREM2 IPCs severely impact microglial survival and ApoE clearance. So targeting these rare and unknown signaling pathways in relation to TREM2 and Alzheimer's disease and exploring them and forming sort of comprehensive study models of these signaling pathways to understand them better can help us understand, help us not only gain a better understanding of the Alzheimer's pathogenesis, but can also give us a better insight into the therapeutics and the future of therapeutics with respect to Alzheimer's disease. Thank you.
Hi, my name is Arjun, and today I'm going to be presenting on my review paper, which is about the role of oligodendrocytes in Alzheimer's. Let's first talk about what oligodendrocytes are. The brain itself is composed of neurons, but by themselves, the brain would not be able to function because neurons need other cells to support them. These cells are called glial cells, and they're astrocytes, microglia, and oligodendrocytes, and they're all responsible for regulating neuron function and helping out with certain tasks. Oligodendrocytes in particular are these large branch cells that sometimes have up to 60 arms that all connect to neurons. Oligodendrocytes are responsible for creating a substance called myelin that overall allows neurons to signal faster, and it's been linked to cognitive processing. Due to this role, oligodendrocytes are vital for proper functioning of neurons and the overall health of the brain. And it only makes sense, therefore, that when oligodendrocytes are disrupted or altered somehow, it would affect the brain, possibly in a negative way. and that is the focus of my review paper. In recent years, more research has emerged that suggests oligodendrocytes play a key role in Alzheimer's and potentially even a causative one. So oligodendrocytes stem from a progenitor cell known as oligodendrocyte precursor cells, or NPCs, OPCs. These precursor cells emerge from the subventricular region of the brain and migrate to regions where they're needed. OPCs can differentiate into myelinating oligodendrocytes if need be. For example, if there was a lesion or a region of demyelination in the brain, these OPCs would then differentiate to compensate for that loss, and this is responsible for synaptic plasticity. Moving on, let's talk about the traditional understanding of Alzheimer's pathology. Traditionally, it's understood as a dual proteinopathy, which means that there are two proteins responsible for causing it, amyloid beta and tau. Amyloid beta first emerges in the entorhinal cortex below the hippocampus. Amyloid beta 42 is the most fibril-forming amyloid species and forms large aggregates that are cytotoxic. These aggregates themselves produce further toxic amyloid beta 42 oligomers that cause synaptic loss and cell death. Drugs that have targeted amyloid beta have shown variability in effectiveness, and many trials have been poorly designed. And it's also hard to get confirmation of these models in animals. So while the amyloid hypothesis has not been validated, it hasn't been fully substantiated either. Tau is a microtubule-associated protein that gives microtubule stability, especially along the axons of neurons. And this allows for axonal growth and other vital properties. When tau is hyperphosphorylated, tau proteins decouple from the microtubule, and this results in insoluble neurofibrillary tangles. And these tangles then affect axonal transport in both early and late-stage Alzheimer's. Now, let's look at the multiple lines of murine model evidence or mice model evidence for OPC involvement in Alzheimer's, beginning with the triple transgene model. So studies noted a significant decrease in OPC proliferation at 6 months, and this is measured by the number of mitotic sister cells, or the cells formed by the mitosis of one cell. And there was also accumulation at amyloid beta plaques. Then in the 5x FAD murine model, there was altered oligodendrocyte precursor cell transcriptional profiles, which suggests altered behavior in Alzheimer's and potentially a role in pathology. They also noted crowding at amyloid beta plaques. Then in the APP/PS1 model, there was decreased oligodendrocyte precursor cell density and increased sensitivity to amyloid beta toxicity. And this was mediated via calcium dyshomeostasis and increased production of toxic A beta forms. Then in vitro studies suggested that amyloid beta induced OPC senescence or aging via 3 factors. P21 CDKN1A, P16 INK4 CDK2NA, and B-galactosidase. Lastly, when they removed a factor that was responsible for cell survival, FGF2, these subset of OPCs known as plexin B3 plus OPCs secreted amyloid beta. And this suggests that when you take away a factor responsible for cell survival, these types of OPCs actually contributed to Alzheimer's pathology. Moving on to the role of myelin. So my paper proposed a cyclical model for myelin and Alzheimer's pathology. So myelin and oligodendrocytes have the highest iron concentration in the brain. So when myelin breakdown occurs, it releases iron and other reactive oxygen species. Iron in the past has been shown to trigger amyloid beta fibrillation, and amyloid beta has been shown to cause demyelination, especially in areas near the amyloid plaque. So my paper suggests a cyclical model, but this model will require further experimental validation. Let's move on to MRI imaging studies that suggest the role of myelin in Alzheimer's pathology. The figure on the left shows a healthy, non-Alzheimer's pathological brain, and the figure on the right shows the comparison between Alzheimer's brains and control brains, and the yellow regions show altered myelin behavior. And specifically, the regions of interest are the cingulate cortex and other cortexes as well. This study was conducted using Q-space myelin mapping. Then moving on to the role of myelin basic protein, which is a protein responsible for myelin stability in the brain. Myelin basic protein is theorized to attach and degrade amyloid beta, which prevents fibrillation. The figure on the left shows the presence of amyloid beta via a thioflavin fluorescence assay with and without myelin basic protein, showing a clear decrease in amyloid beta with MBP. On the right, the graph showed the decrease of two amyloid beta species, amyloid beta 40 and 42, in the presence of MBP. And what this suggests is that MBP, when myelin is broken down and MBP is lost, there is potentially the loss of an agent that decreases amyloid beta presence and prevents amyloid beta fibrillation, which suggests a role of myelin breakdown in Alzheimer's pathology. Then moving on to wild-type mice models, there was notable decrease of phosphatase activity, and phosphatase removes phosphate groups from objects in the white matter of the brain, and this was done with the injection of insoluble tau aggregates. Furthermore, there was a facilitation of tau seeding and prion-like propagation through the brain. So the oligodendrocytes actually move tau throughout the brain. Then in human MRI and PET imaging studies, it indicated the decreased tau uptake in myelinating cortical regions, which could support tau oligomer toxicity and demonstrate how toxicity affects oligodendrocytes. So, in conclusion, Alzheimer's is traditionally understood as a dual proteinopathy of amyloid beta and tau. But new evidence suggests that the demyelination of axons and impaired OPC differentiation and proliferation are characteristics of Alzheimer's as well. Furthermore, there are some studies that have to be conducted to clarify some things. Firstly, studies need to be conducted to determine whether the behavior of OPCs is different near amyloid beta plaques, which could indicate the plexin B3 plus amyloid beta secretion, as well as the role that myelin plays in protecting axons from amyloid beta toxicity, and also validation for the cyclical model of Alzheimer's progression. Thanks for listening.
Hi, my name is John. Today I'm going to talk about using Google search trends to identify regions with vaccine hesitancy. So Google search trend data are samples of actual search requests made to Google. They can be used to describe the popularity of certain Google phrases over time period across different areas. And Google search trends could be used to imply people's interest because, not surprisingly, in our modern world with advanced internet technology, people tend to have the habit to search up things that they are curious about, and in order to learn more about it. So there were already studies investigated the relationship between Google search interest for COVID-19 symptoms and the actual COVID cases. Some study found that the Google search trend could be really effective to forecast the outbreak of the pandemic. On the other hand, some study found that the method was unreliable and it only works for certain phrases. So for the graph on the right, the phrase ageusia has a really high correlation value, 0.817 with the actual COVID cases. But for the phrase the loss of taste, it only has a correlation value of 0.297 with the actual case. One thing we need to notice here is that ageusia means that lost the function of taste, which means the same thing as loss of taste. So the way how people formulate their language to search phrases can really make a big differences. Another key point in our study is the idea of the vaccine hesitancy. The COVID-19 vaccine was first released to the public in US around February 2021, but many communities were hesitant to get the vaccine due to following reasons. First is the religion reasons. The example of it is that the followers of the Christian Science. So they believe that the illness is caused by sin and can only be healed by prayers. And for them, vaccination can amount to sinful rejection of faith in God. So they reject to get the vaccine. Another reason is due to the historic marginalization. So for example, in Bay Areas, African Americans were being used to test the functionality of the vaccine in the past. So currently, many of them reject to get the vaccine. Another reason is due to concerns over the FDA approval process, and there are many reasons along with it. CDC tries really hard to persuade the public especially the hesitant group to get the vaccine, because COVID pandemic will end faster if more people choose to get the vaccine. The main research question in our study is that how does the Google search popularity for phrases associated with vaccine hesitancy correlate to the vaccination rate across several cities in the United States? The method that we use in this study is that we use the RStudio to process our data, including to graph our data and obtain the coefficient correlation value, which is known as the R value. We obtain our vaccination rate data for San Francisco, Houston, Chicago, and New York City for every week in 2021, and sustaining for a whole year. We chose these cities because they are major cities in different parts of the US, such as North, East, South, and West. The Google phrases that we use in this study include, is the vaccine safe? Should I get the vaccine? Vaccination location, vaccine mandate, and also we include a control search phrase banana. The function of it is to measure the sensitivity of our method to detect the spurious correlation. In other words, to test whether our method to perform this study is really useful or not. This is a typical graph that we got from RStudio. So the x-axis is the time series from June 2021 and for a whole year, and the y-axis is just on the relative scale of the count. This graph is for New York City, and the search phrase is the vaccine safe. The blue line is the vaccination doses. We divided all our dose number by 1,000 in order for it to match up with the search popularity. Search popularity can only be a value between 0 to 100, because Google published their data on a relative scale. The way how they form the popularity is that they use the popularity of this specific phrase and compare it with the popularity of all other phrases at the same time period to generate a number between 0 to 100 to represent its true popularity. So graph here shown that for the first peak of the vaccination, which is around February 2021 to July 2021, the lines might match up nicely, meaning that the association is pretty strong and is positive for this time period. But after the first peak, the association became really vague and not obvious. And here is the graph for our control banana. So the trends do not line up at any time, and the search phrase for banana will always sustain a pretty high stable level with some minor fluctuation. But people can still argue that if you look at it closely, there are some really minor association between these two trend lines. Here is a heat map analysis for the phrase is the vaccine safe, which is a really popular phrase in our study. The heat map shows the correlation between the search interest and the vaccine rate over the whole year. And the correlation is calculated over a window of 2 months for each time point. The yellow and more bright color means that there's a positive correlation between the vaccine doses and the search popularity, and the dark color and dark purple implies a negative correlation. If the color is light red and light purple, it means the association is really, really weak. We found similar patterns of colors of association from February to June across all cities. They are all bright colors, yellows and red, which justified what we saw previously that during the first peak of the vaccination, there is a strong correlation. And here is the heat map for a less popular phrase, vaccination location. So the color did not line up any kind here, and meaning that the association is not consistent across cities at any time period. So the main result of our study is that, so for two phrases, is the vaccine safe and should I get the vaccine, two really popular phrases, we found that a consistent positive associations across all 4 cities during the first wave of the vaccination. This could be a really true association or just be a spurious correlation, because it is not surprising that when the vaccine first come out, people tend to search more about it because they want to learn more about it. But on the other hand, people who knew they want to get the vaccine will get the vaccine when the vaccine first come out. So even though the result may be something that we want to see, but the process that we reach our result is not ideal. It's not like people will search up phrases before they want to get the vaccine. The limitation of our study is that the vaccine data and the search trend data encompass slightly different regions and people. So for example, for our search trend data, they are all on a relative scale instead of the raw data. And also the vaccine data, many of them are on a county level instead designated for a specific city, which could cause some inaccurate results. The conclusion for our study is that the method is being proved to be unreliable because only certain phrases can pick up an association, and there are just too many confounding variables involved with it. And even if we did detect an effect here, this is a really high possibility that it is due to the popularity of a specific phrase, the time period, or just due to randomness. As we see that for our control variables, we still see some association with the vaccine doses, even though there should not be any association. However, we still believe that the Google search trend could be potentially used for the vaccine surveillance of different data or methods, such as if we can get the data, vaccine data for a specific city, or the Google search trend data on a raw scale. So even though we did not find a method to be reliable in this study, but we believe an alternative study or approach could use the Google search trend data more effectively. And we highly believe the potential of the Google search trend data because with the advance of the technology, people tend to search Google and use Google a lot to search things that they are curious about. Thank you.
Hello everyone, my name is Haniyah Bilal and I'll be presenting my research on COVID-19 and menstrual health. In particular, the impact of the SARS-CoV-2 infection on menstrual cycles and PMS symptoms in women in Lahore, Pakistan. Now here's just a little bit of an overview of everything that will be discussed today. Now, before I even begin talking about my research, I wanted to take a moment to talk about why research in this area is so necessary, or in this field is so necessary. And that is simply because women for the longest time have been underrepresented in medical literature. A very simple example of this might be heart attacks, and how female symptoms vary from those of men, which is why they go undiagnosed longer, and therefore have higher mortality rates. In fact, according to this research paper that studied various medical research articles, found that female representation was just at 37%. Now, of course, this is also an issue when it comes to studying the female reproductive system, which has less research in it happening when it comes to the field of gynecology and obstetrics, in particular that of gynecology. It does suffer from less research happening in it, which is why it's so important that more research be happening in this area. This is particularly true for women of color, such as those of Southeast Asian descent, which my research will primarily target. Now, why this particular research? I'm researching the effects of the impact of the COVID-19 infection upon menstrual health and women's health, in particular how it's affecting their PMS symptoms and their menstrual cycles. So why this? And the reason for that simply being because, as stated in this research paper, questions about menstruation have long been excluded from most large-scale COVID-19 studies, and that remains true. While we have a lot of research happening that has been published and that is still currently happening underway when it comes to COVID-19 or even long COVID, there is limited research. There is very limited research when it comes to how COVID-19 has affected menstrual cycles and menstrual health of women. However, there has been an increased popularity in anecdotal information regarding menstruation in the COVID-19 infection, which simply goes to show that this is an area or a gap of research that needs to be covered, which I hope to, in some part, do with this research. Now, just an abstract or an overview. is that the study aims to study how the SARS-CoV-2 COVID-19 infection affects women's menstrual cycles and menstruation. It'll look at both how long COVID and regular COVID appear to affect the following: the length and duration of the cycle, changes in the heaviness of flow, potential blood clotting, dysmenorrhea, amenorrhea, all PMS symptoms, and the impact of pre-existing gynecological, the impact it has on dysmenorrhea faced as a result of pre-existing gynecological conditions such as PMDD, endometriosis, etc., just to name a few. Now, long COVID refers to COVID-19 infections that are generally more severe and last over 2 weeks on, generally over a month long, and have symptoms that last afterwards as well. Whereas regular COVID is generally an infection that lasts around 2 weeks and does not have any long-lasting symptoms attached to it. Now, factors that I'll be considering when I look at this will of course be long COVID versus regular COVID and how that's impacting PMS symptoms, length of menstrual cycles, and how pre-existing gynecological conditions, dysmenorrhea, is affected by it. Now, limits that will be set on this is of course that it'll be for menstruating women only, therefore pregnant women cannot be included. It'll be for adult women above the age of 18 simply because puberty and adolescence may affect menstruation in other ways as well. Apart from this, one thing we will be looking at, and one factor that will also be considered, is the COVID-19 variant, and whether it's possible that a particular variant may be affecting the symptoms of PMS and menstrual cycles differently. So the exact COVID variant will also be noted. Apart from this, any medication they may have been on, blood thinners can affect menstrual flow, lifestyle factors, because lifestyle can affect menstrual health, perhaps by causing amenorrhea due to stress. Caffeine and alcohol can exacerbate PMS symptoms as well, according to certain research. Of course, we will also be looking at their vaccination status and any pre-existing gynecological condition they may have had. Now, the focus of this research will be, as mentioned before, primarily on long COVID versus short COVID. We'll look at all the PMS symptoms. Here I've just listed a couple, but we will be looking at the official list of the entirety of all PMS symptoms and how each one of them is affected or not by COVID-19 infections, be it long or short, and regardless of which variant it is. Mainly also focusing on dysmenorrhea, which is menstrual pain, which will be measured by how many over-the-counter painkillers are required to bear the pain, pre-infection and post-infection, clotting, amenorrhea, and length of menstrual cycles. The reason these will be primarily in focus is because previous research is showing that there is a correlation between these factors and COVID-19 infections. So, for example, previous research shows that you have an increased risk of menstrual clotting post-COVID-19 infections. Apart from this, dysmenorrhea has possibly increased post-COVID-19 infections. So this current research is suggesting that we decided later on, is suggesting that 22.4% of women experienced worsened pain of dysmenorrhea post-infection. And dysmenorrhea appears to be worsening in women who've had more severe infections, such as that of long COVID. The duration may have slightly lessened as well, which shows that there's a possibility that post-COVID-19 infections, menstrual cycle may have shortened. Apart from this, there is research that indicates that there may be effect or a change on the menstrual flow, while one paper cites that 18% reported heavier flows or decreased flows, while another reported a drastic increase in lessened flow. So while exactly whether infections are lessening flows or increasing it, there seems to be some vagueness about that, ambiguity about that. But one thing we can know for sure is the possibility that heaviness of flow may be affected by COVID-19 infections. And lastly, of course, is amenorrhea. Because, however, amenorrhea seems to be mainly temporary because most women experience it during the infection itself, but gradually post-infection, the number shifts after a year. Now, the research methodology I'll be using is a Google survey. I'll be surveying using the survey online. Here are just a couple of my sample questions. It will be an online survey with consent forms given and will of course be anonymous. During the start, the first thing you can see here is simply just prerequisites, being an 18 plus menstruating woman. However, apart from this, you can also see how I'll be collecting information. For example, certain PMS symptoms, I'll be looking at them before, during, and after COVID and having them rate the frequency or severity of them. Apart from this, pre-existing gynecological conditions are listed down below, all of them which can be selected, or any others as well. Yes, and same thing applies for menstrual length cycles, etc. Now, while my research is still underway, what I'm hoping to find or accomplish with my findings is to have a deeper understanding, or at the very least, just simply evidence, if not, conceptual understanding, evidence of statistics about how the COVID-19 infection is affecting certain aspects of menstrual health. Now, these include, of course, PMS symptoms, any dysmenorrhea that is associated with pre-existing gynecological conditions, and the length and duration of menstrual cycles, as well as any clotting or amenorrhea that may be occurring. Now, once I have my findings, I would like to ideally compare statistics between of symptoms that have been increased or worsened or affected in any way between long COVID and regular COVID. So I would, of course, like to have that comparison there. I would also like to have comparison between the changes that are associated in menstrual cycles and PMS symptoms or dysmenorrhea amongst the different variants of the infection as well, to see if there are any changes there. And obviously, of course, the PMS symptoms or dysmenorrhea that is being looked at will obviously be compared pre-infection to post-infection as well, just to see what changes have happened. And for pre-existing gynecological conditions, if the dysmenorrhea has been exacerbated or lessened, the PMS symptoms, whether these symptoms have worsened or lessened or stayed the same as well. The statistical analysis of this data will be found, will be analyzed using SPS software. And I've already explained how the data will be analyzed. Now discussion. It is possible that these changes in the menstrual cycles and dysmenorrhea may simply be a result of psychological stress of having the infection. Is possible. One possibility is this, and which is subsequently resulting in amenorrhea and dysmenorrhea, because research does show that stress can cause amenorrhea and can even worsen dysmenorrhea, possibly, or other PMS symptoms. However, another possibility is that the SARS-CoV-2 virus affects the body, also affects and infects the reproductive system. So one thing can be that during more severe COVID-19 infections, due to the stress of being so ill, amenorrhea can occur. Amenorrhea and irregularity can occur simply due to the stress placed on the body due to the more severe infections. However, apart from this, another thing that could possibly be happening is that the SARS-CoV-2 virus is directly affecting the female reproductive system and attacking it, and that may be due to the fact that the ACE2 receptors are directly affected by the SARS-CoV-2 virus, and these ACE2 receptors are frequently expressed and can also be found in the female reproductive system, including regions and organs such as ovaries and the uterus. And due to the role the ACE2 receptor plays within the female reproductive system, it might be possible that the virus is affecting female health in that regard. Well, I hope that my research can at least show some statistics about how exactly COVID-19 infections are affecting certain aspects of menstrual health. And I hope it can also help people later on in terms of understanding how or why these changes are occurring. Thank you so much. Here are the citations. Thank you so much if you've made it through and for listening. Thank you to my family and colleagues for giving me this wonderful opportunity to research, and goodbye.
Hello everyone, today I'm going to present how will internal combustion engine technology for the passenger car evolve in the era of electric vehicle to meet tighter emission regulation. My name is Albert, and a big thank to my mentor Adrian, who helps me a lot on my project. So first of all, what are internal combustion engine vehicles and electric vehicles? Internal combustion engine is an engine that generates motive power by burning of gasoline, oil, or other fuel with air inside the engine. The hot gases being produced drives the piston or the other work as they expand. And electric vehicles are vehicles that uses one or more electric motors for propulsion. And despite the fact that they are rapidly developing, the internal combustion in vehicles still dominates the vehicle, like the passenger car market. And currently in the world, there are approximately 1.2 billion internal combustion engine vehicles right now, and which is huge comparing to merely 16 million of electric vehicles. And when we do some math, we can find out that there are nearly 99% of internal combustion engine vehicle that takes the market share of passenger car. On the other hand, ICE vehicles also have a much longer history compared to electric vehicles. And we can draw the conclusion that it has a rather mature technology compared to a rather emerging technology of electric vehicles. So what are some hurdles of EV to mass adoption? Well, first of all, there are charging availability issues and also driving range of vehicles, especially in winter when the weather is extremely cold. And also there's cost of vehicles comparing to its internal combustion engine vehicle counterparts that cost much lower. And here's a diagram that shows the global long-term passenger car vehicle sales. And by 2040, ICE vehicle is projected to still take up more than 50% of the passenger car market share. And before that, the engine technology will be used to achieve efficiency and reduce pollution. So how exactly can engine increase efficiency and reduce pollution? So we all know that the internal combustion engine energy comes from combustion, and there's three elements of combustion: oxygen, fuel, and heat. And the major challenges of ICE will be incomplete combustion and poor air-fuel mixture. And some possible solutions are, first of all, faster combustion. Secondly, better air-fuel mixture. And thirdly, wasted energy reuse. So I'm going to present 4 of the major technology that help the SE to overcome problems such as incomplete combustion and poor air-fuel mixture. And throughout my project, I also built a mini engine kit that helps me to understand engine deconstructions. And here's a picture that shows the engine after it is assembled. So first of all, compression ratio. For the picture on the left, we can see that the compression ratio is measured as the total volume of V1 plus V2 divided by V2. V2 is the volume marked red, and the lower the V2 is, the higher the compression ratio is, according to this formula. And in real life, the lower the V2 means the better compressed air-fuel mixture by the piston. And therefore, high compression ratio means better compressed air-fuel mixture. And it led to instant and complete combustion of the engine. So then why every car just can't have crazy high compression ratio and then have better efficiency? So it's because they're uncontrolled ignition due to high temperature of compression, and then it will eventually lead to engine damage. And there are also some requirements for high compression ratio. For example, high octane fuel, that usually means higher cost. And on the right, here's some typical examples of engines with high compression ratio. For example, Nissan's VCT engine with a compression ratio of 14:1, and Mazda's HCCI technology enables a maximum compression ratio of 18:1. That that makes engine 20 to 30% more efficient compared to its previous design. And secondly, I'm going to introduce ignition system and typically introduce plasma ignition technology and compare it to traditional spark plug. So on the left, we can see the traditional spark plug with a single heat spot that usually cause the problem of incomplete combustion. And when we look at the plasma ignition technology, we can see that the plasma will ignite the air-fuel mixture in multiple from multiple spots, so that it can enable ignition with less fuel and also have faster ignition and combustion, and also allows high compression ratio at the same time. Therefore, the plasma ignition technology boosts engine efficiency by 20% and cut emissions by 50%. Thirdly, it's the fuel injection. The fuel injector can control air-fuel ratio, and optimum air-fuel ratio provides high efficiency when just enough air and fuel can provide the power needed. Basically, there are two types of fuel injector. The first one is port injection, and second one is direct injection. Port injection guarantees a good air-fuel mixture at low speed. It's because it was injected into the engine with air intake, and it was placed in specific angles so that it can properly mix with the air to form a mixture. And for the direct injection, it guarantees high compression ratio and good mixture at high speed. It's because it is placed atop of the piston, and while the piston is moving really fast, it can guarantee accuracy of fuel injection. And then there's dual injection, which is the combination of port injection and direct injection, as the picture on the right shows. And it basically make the port injection and direct injection complementary, and it achieves flexibility of controlling air-fuel ratio, and that guarantees high combustion speed and also high fuel efficiency. Lastly is the turbocharger. The turbocharger uses energy from the flow of exhaust gas to compress the intake air into the engine. And in this chart, we can see the exhaust gas in red current coming out from the piston, and then in red and then blue intake air being sucked by the rotating turbo and like compressed into the piston. And because there is circulation of currents, there will be a turbo lag caused by the time difference between the exhaust gas leaving the engine and the rotating turbine, and the time that the air was sucked inside the engine. So there's a all-time drawback of the turbocharger. And then here's a new design of turbocharger recently called e-turbo, which is an e-turbocharger. And e-turbocharger harnesses the excess energy to generate electricity, which is stored in the battery right here. So basically, it does not use the energy from the exhaust gas to rotate the turbo. Instead, use the electric energy from the battery, so that it will produce instant boost of the engine from the engine. And then the turbo lag will be no longer a problem, because the turbo is not rotated by the exhaust gas anymore. And overall, the turbo will all generate more power and torque for about 15 to 25 or even more. And then it will also make the engine more efficient by allowing smaller engine that produce the same horsepower. In conclusion, the internal combustion engine technology gives vehicle higher efficiency, which allows them to remain existing and meet rigid emission regulations before mass adoption of electric vehicles. And here's a study that predicts the emission targets in 2013, 2014, and 2050. And we can see that from the passenger car market, we can see that some of the vehicles actually achieves the goal in 2040. For example, Ford Fiesta or Mazda 3. And it again shows that the internal combustion engine has the potential to be more efficient and also exist for a longer time than people expected. That's all for my presentation. Thank you.